We work in the field of mechanobiology. What's mechanobiology? Well, basically it's the way that cells and tissues interact mechanically with their environment. This can happen in a number of ways. One way in which they can do it is by pulling on it. So cells grow on surfaces and within tissues, and as they do that, they feel what's around them. They mechanically interact, they tug on the material, and by doing that, they can find out information about where they're living, what sort of environment they're living in.
You might know the story, The Princess and the Pea. So the princess didn't sleep well because she felt the pea under many mattresses. In the same way, cells can sense the rigidity of a material under soft hydrogels. As the princess felt the pea, the cells are contracting the material and they can sense a rigid object under soft hydrogels.
This can have really profound effects on what the cells do. It can affect how they move around, for instance when we develop. It can affect how cells and tissues move during healing events, and it can also tell cells what types of cells are turned into. For example, stem cells can turn into cells as diverse as bone and nerve just by sensing how soft or hard the material that they feel is.
To find out if cells are able to sense through materials, I've been culturing human cells on synthetic substrates. These substrates are made of polyacrylamide, and we've been culturing different types of human cells. And we've been able to find that groups of cells, they can sense through materials further than single cells. In order to measure how the cells move the gel, we've been using a technique which is known as traction force microscopy, but we didn't measure the force, we only measured the displacement. And in order to do this technique, I prepare polyacrylamide gels with fluorescent beads, and then I culture cells on these gels. And I've been using a microscope to take images in bright field to see the cells, and in fluorescence to be able to see the movement of the beads, which are a result of the movement of the cells. And then I've been using software which runs in MATLAB, which is able to quantify the displacement that the groups of cells produce.
So why is this important? Well, in normal development, tissues are able to feel the mechanical context of the environment in which they live. During disease processes, for instance, one example being cancer, but there are lots of others, the mechanical properties of the tissues change, and that can change the way cells behave. It can make them perhaps more invasive and more disease-like. So it's obviously very important to understand how cells are able to sense these mechanical properties. Another area we're interested in is wound healing. The cells on the surface of the skin have to migrate, move over an underlying layer of tissue. And it's known that the mechanical properties of that material are very important in telling the cells how to arrange themselves and how to behave. So we think that by modulating stiffness and by understanding it better, we can improve therapies which might promote, for instance, wound healing.